section is 35.2 and 35.3, and it's on adaptive immunity. So remember last time we talked about one of the branches of the animal immune system called innate. So the innate immune system is broad, differentiates self and non-self, and it's fast-acting and short-term. We're talking about the other branch, the adaptive immune system, that only vertebrates have. So only animals like us have an adaptive immune system. So one important difference is the adaptive immune system is specific. Remember, the innate immune system can only really identify something's foreign, doesn't belong in the body, or something is body organs. Can't really differentiate about what foreign object it is, but can only differentiate that it doesn't belong in the body. So recognizes, for example, the flagellin protein that is only on bacteria. The adaptive immune system can differentiate and respond accordingly to different pathogens. So it is specific. And the primary actors in this response, it relies on T and B lymphocytes, which are types of white blood cells. These lymphocytes, the T and B lymphocytes, originate in the bone marrow. Just like all of your blood cells, it's made in the bone marrow. So why some people have bone marrow transplants is they aren't able to produce enough blood cells. B lymphocytes stay there and mature in the bone. That's why they're called B cells. They mature in the bone. And T lymphocytes, the other part of the adaptive immune system, migrate and mature in the thymus. That's why it's called a T lymphocyte. And these cells react with bacteria and viral pathogens. And they do that by reacting to substances called antigens. So B and T cells have antigen receptors, so they look kind of like this, proteins on their plasma membranes, that bind with pathogenic antigens. And pathogenic antigens are like membrane proteins. And the part of the antigen on the pathogen that ends up reacting or binding with these receptors is called an epitope. Alright, so here's an example of a B cell. Here is the B cell receptor reacting with the epitope of the pathogen. Same thing, here's a T cell with its receptor reacting with an epitope of this other pathogen. So first we'll kind of talk about B cells. And B cells have antigen receptors that are shaped kind of like a Y. Here's the antigen receptor. You can kind of see how it is a Y shape. And they're composed of two protein backbones, a heavy chain, it's the long one, and a light chain, this short one. So if you see in a bigger picture of a B cell, you can see the heavy chains right here, light chains right there, to make that Y-shaped receptor. And the shape of this receptor corresponds to a specific antigen. So important, every single receptor on a given B cell will be the same or fit with the same antigen, same epitope. So each different kind of B cell can only react with one antigen. We can make hundreds of different B cells that have different receptors, but each receptor in each B cell can only connect with one type of antigen. So when a B cell receptor does bind to the antigen that fits in there, it activates the B cell, and the B cell then goes through rapid mitosis and differentiates into plasma cells. And what these plasma cells is secrete antibodies. So they basically secrete things that look like this, but they're not bound to the plasma membrane here. They're just floating around free in the cytosol. And these end up attaching to those bacteria and those viruses and surrounding them. And one of the things they do is mark them or serve as a signal to tell phagocytic cells to eat them or to destroy them. So here we go. We've got a B cell, and it's attaching, and it causes that B cell to turn into, or to differentiate into plasma cells, which then secrete these antibodies that can attach to any of these antigens. Well, you'll notice is that this receptor is made up of a constant region, so this never changes, or a variable region. The part that attaches to the epitope is the variable region. That changes depending on what antigen it needs to bind to. So different B cells can have different receptors with different variable regions. T cells work much the same way, except they're composed of one alpha chain and one beta chain. Still have variable regions. These are different depending on the T cells. Different T cells for different antigens. And T cells, instead of binding directly to pathogens, they bind to epitopes of pathogens that are presented at the surface of what's called APCs, or antigen-presenting cells, such as phagocytes. So for example, a phagocytic cell might eat or digest, destroy this bacteria, and break it down into little pieces. And these antigen fragments, these proteins from its plasma membrane, get picked up by what's called a MHC, or major histocompatibility protein, and together they float to the surface and are presented on the surface of this macrophage after it digests this. That is what the T cell's receptor will bind to, not directly onto this pathogen, but to the epitope, or the antigen being presented via the MHC domain on this antigen presenting cell. So that's how the T cell becomes activated. So T and B cell light chains, so going back here, these light chain parts, the light chain parts right here, are made up of 3 different genes, or encoded by 3 different genes. A constant variable and joining region, a C, V, and J region. So in their DNA, C, V, J, C. Each T and B cell could have one C, a choice between any 5 different J and any 40 different V regions, as well as, and that's just for the light chain, there's even more options for the heavy chain. So depending on when we're splicing this RNA, when we're splicing it, we can create many different receptors from the same genes. We can create 3.5 times 10 to the 6th, 3.5 times 10 to the 6th different specific antigens, more than enough to respond to any pathogen out there. And those receptors that end up that match pathogens. So this happens very, very, very young and indiscriminately. We'll just match these different things up. However, when you're young, just out of chance, some of these receptors can end up binding to proteins or antigens present on your own blood or your own organs. Now, when they're first developing new T and B cells, your body has a process of, through the lymph system, destroying those self-recognizing receptors. And that's why your immune system is called self-tolerant. It doesn't elicit an immune response against itself. People with autoimmune disorders, though, that doesn't necessarily happen. So when a B cell antigen receptor binds to an antigen, it undergoes rapid mitosis. And that rapid mitosis produces many plasma cells that then secrete antibodies for that antigen. So now they're binding to all of those different viral or pathogenic bacterial cells. Some of them turn into plasma cells that fight the current infection, but some remain as B cells, and they are called memory B cells. Memory B cells. And what memory B cells do is they hang around and are dormant till later, till maybe years later your body's infected with the same virus or bacteria, the same antigen. Then there's already memory B cells stored up. They can start making plasma cells. They can respond much faster. And that's why usually you don't get the same disease twice, because you have these memory cells stored up that tells your body how to produce those antibodies much faster. So for example, the first time that I'm exposed to antigen A, it takes me a while to produce enough of those antibodies to fight off the infection. Then as the infection subsides, I still have a layer, an amount, or concentration of memory B cells still in my body. So then when I get exposed again, all of a sudden I rapidly produce in a much shorter time a lot more of those antibodies. That's why when we get sick a second time, we fight it off and we don't even know we were sick. That's why we usually say we can't get that same strain of the disease twice. All right? And that's because we have these memory B and later memory T cells that expedite our immune response subsequently. T cells are similar, but there are two types of T cells, helper T cells and cytotoxic T cells. So a helper T cell is what binds to those APCs, those antigen-presenting cells. So, for example, maybe this phagocyte eats this bacterium, and its MHC molecule grabs this antigen and presents it, and a helper T cell recognizes it. When it matches and recognizes it, it releases what's called cytokines and interleukins. They're messengers. And what they do is they activate B cells, so even the B cells don't have to bind onto this antigen necessarily. They can be activated by these helper T cells. That's why they're called helper. They're helping out the B cells, and the B cells can activate what's called the humoral immunity. So B cells and plasma cells that secrete antibodies are part of the humoral immunity. And helper T cells also activate what are called cytotoxic T cells, cell-mediated immunity. What cytotoxic T cells do is they destroy infected cells. So if this bacteria would infect one of my cells, it kind of limits, it destroys that cell. So a cytotoxic T cell is looking for the body's own cells that have been infected. So when it binds onto a body cell that's presenting a foreign antigen, kind of like this a phagocyte is presenting a foreign antigen, some of your body cells can do that too. When your body cells present a foreign antigen, it attaches there and it releases perforin, which puts holes in the plasma membrane, and then granzymes that burst the cell. All right? And some of those cytotoxic T cells will also linger after the infection and form memory cytotoxic T cells, much like memory B cells, so you can have a faster cell-mediated response later on. So here's a picture of a cytotoxic T cell binding to an MHC complex presented by one of your body cells. It releases perforin, drills holes, makes pores, and granzymes that then lyse the cell, so limiting the bacterial or the viral propagation by destroying the infected cells. So here's kind of a review of adaptive immunity. First time we're exposed to an antigen, that antigen will cause the corresponding B cells to proliferate and form both memory B cells for later and plasma cells to secrete antibodies. It also will activate phagocytes, part of the innate immune system, that are antigen producing cells. Those activate the helper T cells that then will again stimulate production of B cells, stimulate production of cytotoxic T cells, will fight off the infection. And if you're ever exposed later, a second exposure, your memory B cells will directly start telling you to create more antibodies, directly telling you to activate cytotoxic T cells so you can fight off that response much quicker. This is where vaccination comes in. So vaccines are like injecting you with a virus. It's a weakened or a killed form of the pathogen. And the idea is your immune system, your B and T cells, will react to that dead antigen or that weakened antigen like a regular attack. So start making plasma cells, start making cytotoxic T cells, and it will get rid of the infection super quick because it's not very strong, and you won't even notice it. But your body now has those memory B and memory T cells. So if you get infected again, your body is so well prepared you don't even get sick. So usually you don't even notice the subsequent infection. So it is training your immune system to pretend like you already got infected with that disease but you suffer no ill effects from it. So that's why it's very important to get your vaccinations, because if some of these diseases, if you don't have a vaccination, your immune system isn't prepped for it, it could do a number, and you could end up in the hospital or worse. That's why it's very important. Your immune system also has some unfortunate roles to play, like if you're going to get a transplanted organ. So your immune system recognizes all of the antigens on your organs, but not necessarily others. So that's why others have to take drugs to suppress their immune systems to keep their adaptive immunity and their innate immunity from trying to destroy their new transplanted liver or kidney, because your body thinks it's a foreign invader and is trying to kill it. Also, in allergies, your allergies happen because your body is overly or reacting too much to things like pollen or dander or food, like it's a foreign invader. It's going to start your immune system going. Those mast cells, like from the innate immune system, are going to start secreting histamine. It'll start creating dilation. Epinephrine, adrenaline is going to be secreted to help you fight off or help your immune system, and it's going to create those symptoms like you're sick. So that's why allergies is basically an immune hypersensitivity. Lastly, I want to talk about a subject called autoimmune diseases. So sometimes, like I foreshadowed, an individual's immune system isn't completely self-tolerant. Sometimes it thinks part of your body is a foreign invader, and it will be active against parts of your body. So for example, in this case, the immune system attacks part of your body as if it was a foreign pathogen. So normally, an antigen invades, your antibodies produce and kill the invader, and antibodies remain to protect you. In an immune system, in autoimmune diseases, sometimes the immune system forms antibodies to yourself. Those aren't killed off the lymph nodes like they're supposed to. The antibodies start attacking your own antigens on your organs and tissues, and inflammation and tissue damage can occur. Some things like systemic lupus erythematosus, your body attacks DNA histones, diabetes, your immune system attacks the beta cells of the pancreas, MS, the myelin sheaths of your neurons get attacked, myasthenia gravis, the acetylcholine receptors in your brain and in your synaptic junctions get attacked, and rheumatoid arthritis, your synovial fluid in your joints. And women are more likely to develop autoimmunity than men are, and it's unknown why that is. There are many types of autoimmune diseases, and for a lot of them, we have no clue why your immune response is acting like that. So, active area of research, I behoove you. If you are interested in immunology, you can make your career diagnosing and curing autoimmune diseases, because they represent a huge problem of we don't know why our immune system, we have ideas, but not a lot to help, all right? Right, so if you have questions, let me know. I'll see you on Friday.